here I'm going to take a look at 6 different issues that have to do with neuroplasticity. And the first is neurogenesis, or the birth of new nerve cells in the human brain. And one of these places occurs in the olfactory system. You see here, this is the olfactory nerve. It sends down nerve fibers and receptors down into the nose, and this is where you smell. And what happens oftentimes when people have a closed head injury is that the brain is really shaken badly within the skull. And what happens is it oftentimes will shear these nerves off. And so it's not uncommon that people have a moderately severe head injury. They'll lose the ability to smell. This is oftentimes experienced as I can't taste things. But taste and smell are really intimately intertwined. It's really smell that's damaged. But within 4 to 6 months, almost always, these nerve cells regenerate and you can smell again. Okay, so that's one example. That's not especially relevant to what we're going to talk about today, but that's one of the 2 areas where you can regenerate new nerve cells. But the other, thank goodness for this, is the hippocampus, this limbic system structure. And, if someone were to ask me, if you're going to lose part of your brain, which part, would you really want to hang on to? I'd first say the brainstem, because I would like to continue to be able to breathe, stuff like that. But maybe second to that would be the hippocampus, because the hippocampus is critically involved in 2 things. Now, one thing's been known for decades, and that is the role the hippocampus plays in learning. It's the ability to encounter new things and put it to memory, and eventually it gets consolidated into longer-term memory. And if you have damage to the hippocampus, and this happens in Alzheimer's disease and many other disorders, people can't learn new things. And to navigate through life, you've got to be able to do that, really, or people are very impaired. The other thing that's a more recent discovery, though, is the role of the hippocampus in controlling emotions, affect regulation. We're going to talk quite a bit about that, okay? And as I said a moment ago, thank goodness, it's been discovered that this is the other part of the brain that has the capacity for repairing itself by way of growing new nerve cells. And this is a big deal, okay? And we'll come back to this a number of times today. Next is dendrite proliferation, and the little neurofilaments coming off of the cell body, dendrites, grow. And some dendrite growth is programmed early by genetics, but the vast majority is actually influenced by life experiences. And so if you actively use a nerve or a neural network, a group of nerves together, one thing that happens over time is dendrites grow. And it can be influenced by experiences. Also, it's dependent on the availability of nerve growth factors. And this is a class of proteins. Now, if you'll drop down here to the bottom bullet point here, BDNF, this is brain-derived neurotrophic factor. We'll talk about it a lot today. It's certainly not the only nerve growth factor, but it's maybe one of the more important ones. It's the one that's been researched the most. And these nerve growth factors, including BDNF, can be activated by exercise. Okay, so you can actually increase the capacity for the brain to grow new dendrites by exercise, and also by some psychiatric drugs. This is a neat slide here, a nerve cell in the laboratory, and then they inject into the fluid surrounding the nerve cell BDNF, and look what happens in 24 hours. Massive growth of dendrites. So when you talk about repairing the brain, in selected areas of the brain you can actually grow new nerve cells, but throughout most of the brain the repair is largely due to increasing synapses and growth of dendrites. And this can be a big deal. Let's take a look here. This is a brain map showing some fairly specific areas for, this is the motor area, and this is in the parietal lobe here that we're going to look at, and this is a tactile perception. So when I touch something, most people know that most of the sensory experiences on the left side of the body are represented in the right hemisphere, most of them. Okay, so when I touch the podium here with my left index finger in the right hemisphere, it would light up. It would be metabolically registered in the area designated for the fingers here in the parietal lobe. Now, if you do something over and over and over again, the part of the brain grows, okay? And it's mostly due to increased proliferation of dendrites. And in this interesting study that was done 10 years ago by Pascal Leon, these are people that were learning Braille. These were newly blinded people, okay? And they decided they would learn Braille, which I think is really amazing that people can do this. And so what they did, there are ways of doing brain maps that's non-invasive using craniomagnetic stimulation, so they could determine exactly that part of the brain, how big it is for the index finger. And then you learn Braille with your dominant index finger. Okay, so what they did is they got into this study. They did a brain mapping before they started, and then you can see on the slide 2 hours a day of class, 5 days a week. And by 6 months, they had learned how to read Braille, but when they went back and did brain mapping, that part is designated for the index finger, has significantly increased in size. Okay, so we're not talking about just transient changes in neurotransmitters or chemistry of the brain, but literally sculpting the brain, changing the structure, morphology of the brain. These are identical twins, separated at birth. They're now 21 years old. These two women, and they have identical DNA, so they really look just almost exactly alike. But look at their brains. I don't know how well you can see this or not, but these brains are not the same, okay? These are brain scans, and look here at the position of certain fissures and gyri. They are clearly different. And so even not at a microscopic level, you can say, boy, these brains are different, despite the fact they have identical DNA. And it's because life experiences are different, and it hugely impacts the development of the brain. Throughout life, within minutes, synapses can be created or eliminated. So let's take a look more specifically at what happens here. This is a diagram of a nerve cell, textbook kind of diagram here. These are dendrites coming off of the cell body, and on the surface of dendrites are dendritic spines. Now, in human beings, the vast majority of synapses, connections between two nerve cells, occur between the terminal end of a nerve and a dendritic spine. You can have synapses going right into the cell body and that sort of thing. Most of them are these connections between spines and nerve terminals. And what can happen is within minutes, spines can grow. And within minutes, spines can disconnect from the synapse and shrink, and you break the synaptic connection. This is photographs here. This is a dendrite. This is an existing spine, and look here, within a half an hour, it's grown a completely new spine. We're not talking about, a matter of weeks or months. We're talking minutes to develop. How do they do that? The production of nerve growth factors when they get stimulated in particular kinds of ways. Now, this is something I want to talk about very briefly. I wish I had more time, but when you get in close proximity a spine and a nerve terminal, they're floating around in this fluid, and it's very unstable. And the way you stabilize the synapse is by the laying down of this connecting stuff here. This is referred to as NCAM, neuronal cell adhesion molecules. And it's kind of like if you have a big ocean liner and it's tied up to a dock, and it's got maybe 30 different tie lines that kind of secure it to the dock. So there's still a space in between here, but it stabilizes the synapse. Now, the thing that I'm going to talk about just very briefly is that when people, and of course other animals too, experience extremely severe stress, there can be a huge surge of the stress hormone cortisol. And what cortisol does is many things, what we're going to be talking about. For most people experience like a single event trauma, this surge of cortisol won't last very long. It'll come back down. But it may be there long enough for it to cause problems. And one problem is it sets in motion a process where another molecule called PSA is increased. It comes in here and dissolves NCAM. And this especially hits hard at the hippocampus, the part of the brain that is so important in taking in new information and remembering this. So some people that have amnesia following a traumatic event, a part of this can be actually neurobiologically based, because within half an hour or so you can have the disconnect of hundreds of millions of synapses in the hippocampus, rendering it unable to really store the memories of the traumatic event. And in fact, when people have PTSD, if they have only really vague memories or they're amnestic for it, they actually have a much higher risk of developing PTSD. Okay? So this is just an example of how a stressful life event can destroy synapse. And when you destroy NCAM, then the spines begin to shrink back down into the dendrite, and you've lost the connection. Reassignment. This is the notion that certain parts of the brain, if they get badly damaged, then it's possible to some extent for another part of the brain to take over their function. Okay, an example of this is in cats. If you are able to measure their brain activity, you'll find that when they get visual stimulation, just normal visual stimulation, you also see stuff lighting up in the tactile area. So there's already some connections in a different part of the brain that are registering visual input. If you blindfold humans for 5 days and then you touch them, they not only light up metabolically in the tactile perception area, but by 5 days their visual area starts to light up too, which is very interesting. And also, and this is something we'll come back to several times today, and that is, even in the absence of external stimuli, you can have, any of us can close our eyes and we can imagine something, of whatever, a visual image, and that will light up the visual cortex. So it's possible by paying attention to certain things and using imagery to activate parts of the brain, even in the absence of actual stimulation occurring from the environment.